Let us see another means of direct reversal of DNA damage, and that is by de-alkylation. You know, alkylation of DNA causes the mutations. So there are mechanisms for direct de-alkylation. Direct de-alkylation reactions have probably been most extensively studied in Escherichia coli. Three different proteins can catalyze the direct removal of alkyl groups attached to oxygen atoms in the DNA. So the alkyl groups can attach to the different oxygens present in the DNA. Most probably these oxygens are of nitrogenous bases. So they cause the alteration in the nitrogenous bases and ultimately leads to the mutations. So these three types of proteins include O6 alkyl guanine DNA alkyl transferase 1, O6 alkyl guanine DNA alkyl transferase 2, and the third one is alkyl guanine DNA alkyl transferase. So these three enzymes or proteins, they are involved in this de-alkylation reactions. So first is the O6 alkyl guanine DNA alkyl transferase 1. So this enzyme, that is O6 alkyl guanine DNA alkyl transferase 1, can remove methyl and other alkyl groups which are attached to the O6 atom in guanine. This enzyme can directly remove these groups. It can also remove alkyl groups attached to O4 atom of thymine and to phosphotriesters. Sometimes the phosphate groups, they get alkylated and they are converted into phosphotriesters. So they can also repair these phosphotriesters. This enzyme is a monomer, and it has a flexible linker. It has two domains. One is N-terminal domain and the other one is C-terminal domain. And these two domains, they are linked together through a flexible linker. So there is a flexible linker that connects its C-terminal and N-terminal domains. Each domain has an active site that performs a specific function. So this is the diagrammatic representation of this enzyme, and in this enzyme you can see this is a C-terminal domain, this whole one is C-terminal domain, and this is N-terminal domain. So this one is a flexible linker between these two domains that links these two domains. The N-terminal domain transfers an alkyl group from an sp phosphotriester to one of its own cysteine residues, that is cysteine 69. So there is a cysteine residue present in the N-terminal domain, and this cysteine is cis 69. So this enzyme can directly transfer alkyl group from phosphotriester to its own cysteine group. Similarly, the C-terminal domain transfers an alkyl group from either O6 atom of alkyl guanine or O4 atom of alkyl thymine to one of its own cysteine residues, which is cysteine 321. So same as N-terminal domain does, the C-terminal domain does the same. That is, it removes alkyl group and transfers to its own cysteine residue. But this cysteine is different. This is cysteine 321. So here you can see both these reactions. So in this reaction, this one is methyl phosphotriester. Here you can see a methyl group has been attached to this phosphate group, and now this phosphate has formed three ester bonds. One with this, the other one is this, and the third one is this. So this is a triester. So this one is the enzyme which will reverse the damage, which will repair this change, and it can transfer this methyl group to its own cysteine residue. Similarly, on this side you can see this is O6 methyl guanine. This is altered base, and in this base one methyl group is present to the 6 oxygen of guanine. So in the similar way, the C-terminal domain, it transfers this methyl group to its own cysteine residue. So here you can see this methyl group has been transferred to cysteine 321, and while in this case the methyl group has been transferred to cysteine 69. So in this way, this O6 methyl guanine is converted into guanine, and this phosphotriester is converted into phosphodiester. So in this way, this enzyme repairs the DNA damage. Once alkylated, the protein cannot be regenerated. This enzyme cannot be regenerated and therefore behaves more like an alkyl transferring agent than an enzyme. So now once it has received the methyl groups from DNA, it cannot anymore work as an enzyme. Such proteins as O6 alkyl guanine DNA alkyl transferase 1, which lose their activity after acting one time, are called suicide enzymes. So because once they remove the alkyl group, attach that alkyl group to their own residues, they become functionless, and these enzymes are called suicide enzymes. O6 alkyl guanine DNA alkyl transferase 1 has one additional remarkable function, and that function plays an important role in the metabolic regulation of these enzymes. So after methylation at cis69, this protein is converted to a transcriptional activator. This activator stimulates the transcription of the gene that codes for it, as well as some other genes that code for the proteins that repair DNA damage. So once this methyl group or any other group has been transferred to cis69, this enzyme is converted into an activator, transcriptional activator, and this activator stimulates the transcription of which gene? Of the same gene which is responsible for the synthesis of the same enzyme. So it is a sort of auto activation, because the product of this gene after alkylation is activating the same gene, and is also activates the genes of some other similar proteins. This additional activity permits the bacteria to adapt to environments in which they are continuously exposed to alkylating agents, by synthesizing more copies of these enzymes which can repair damaged DNA. So there are some situations where there is continuous alkylation of DNA. So there must be continuous production of these enzymes. So this mechanism, this metabolic regulation, ensures the continuous production of these enzymes, so that the continuously damaged DNA can be repaired. O6 alkyl guanine DNA alkyl transferase, that is synthesized after all alkylation damage has been repaired, will remain unmethylated, because all the damage has been repaired. So there is no alkyl group which will be transferred to the enzyme. So it will remain unmethylated. This unmethylated form of the enzyme blocks transcription of the same gene that were activated by the methylated protein. So it acts as a sort of feedback inhibitor, and it stops the further synthesis or further expression of the same gene. So in this way, these enzymes regulate themselves.